With chapter 3, we're really getting into bioenergetics and overall metabolism. Keep in mind on here, the primary, the three energy systems and really how they're all working together. Because really, what overall metabolism is trying to do is trying to produce enough energy, that is ATP, to supply the neuromuscular unit. Because remember, we learned in chapter 1, neuromuscular units where power is being developed. Metabolism then is supplying the energy to keep the neuromuscular unit going. And also, in a lot of ways, cardiovascular system we talked about too, that's helping to keep metabolic system going as well, which then allows the neuromuscular and so forth. So all this is all intertwined and connected with one another. So really pay attention to the energy system. You should know these since you've had XPhys, but now I'm going to throw my twist on there. So those who didn't have me will hear my thoughts and what I think about that in terms of strength training. So just really make sure you know these. We're not going to know them as in depth as XPhys, but still I want you to have a pretty good understanding of metabolism, bioenergetics, and overall energy systems. Getting into chapter 3, bioenergetics of exercise and training. Some key definitions. 1, bioenergetics. The flow of energy into a biological system. The conversion of macronutrients into biological usable forms of energy. So bioenergetics is basically looking at just energy production itself. Because then you get the catabolism and anabolism. That is, catabolism, the breakdown of large molecules into smaller molecules, associated with the release of energy. Anabolism, the synthesis of larger molecules from smaller molecules, can be accomplished using energy released from catabolic reactions. Then you have exergonic, energonic. Exergonic reactions being energy releasing. They're generally catabolic. Endergonic reactions require energy and include anabolic processing and contraction of the muscle. So all this combined, looking at all the reactions taking place, both catabolic, anabolic, and anabolic, all of that with the flow of energy, that's what metabolism is. And this is a total of catabolic or exergonic reactions in a biological system. So the difference is when you're looking at this is bioenergetics is looking at just really the flow of energy, how ATP is being produced, both like you see the definition down there, how energy is transferred via exergonic to energonic reactions. So bioenergetics is looking at just overall how ATP, the kinetics of ATP, how it's produced from the macronutrients, fats, carbohydrates, proteins. Where metabolism then is looking at all of it combined, looking at all the different reactions taking place. So a lot of times people use them interchangeably, and I mean, they, for our purposes, kind of we could use them interchangeably, but really scientifically, one's looking at just the flow of energy, while metabolism is looking at all reactions occurring. Bioenergetics is a part of metabolism, but not the other way around. The structure of ATP, we should kind of know this all by now, but it's an adenosine group, and then triphosphate, so it's got 3 phosphates attached on it. And it says a high energy chemical bonds. So phosphate, when anything gets a phosphate placed on it, it basically becomes activated. It becomes a high energy at that point. Okay, creatine phosphate, we'll talk about that coming up. Creatine by itself is really not much, but when you put a phosphate on it, it becomes activated. ATP becomes activated when you have that 3rd phosphate placed onto it. Okay, so it's just a high energy molecule made up of, like I said, the adenosine group with 3 phosphates. It says the hydrolysis of ATP breaks the terminal phosphate bond. That releases energy and leaves ADP and an inorganic phosphate and a hydrogen ion. So a lot of times when we're talking about ATP breaking, we usually say ATP hydrolysis. We use that term because you can see on there that H2O, that water, is a part of the reaction. And when it breaks apart, you're left with an ADP and then the phosphate. And this is hydrolysis of ADP breaks the terminal phosphate bond, releases energy, and leaves AMP, phosphate, and hydrogen. So adenosine monophosphate. And so that's kind of just the flow, the structure of ATP through all of that And so it's just that breaking apart, the release of that phosphate that's given off, then you see energy. And I know in chapter 1, I talked about 75% of ATP use is at the cross-bridge cycle. Well, also with that, pretty much 75%, or pretty much a majority, over around 75% of all ATP usage across the board is just given off as energy, given off as heat, I should say. Only about 25% of ATP use is actually used as mechanical work. So most of the ATP that's being broken apart is just to release heat. Well, when it says energy right there, it's given off as heat. So just kind of know the terms on here for the sake of this course. For this chapter, we're going to be looking at more or less what happens when you have a buildup of phosphate, buildup of ADP, AMP, hydrogens. That's the important kind of takeaway from this. You have 3 basic energy systems. You have the phosphagen system, ATP, PCR. For those who have me for X-Phys, I call it the immediate system, and I'll kind of show you why coming up here in a bit. You have glycolysis, and you have oxidative system. Okay, I'm going to bring this up right now. This is one thing that I kind of have a problem with, but a lot of times people talk about, you'll see it, even the book talks about it, fast and slow glycolysis, aerobic and anaerobic glycolysis. Those are kind of misleading terms, because glycolysis, defined as, is the breaking apart of a carbohydrate into pyruvate. The carbohydrate being either glycogen or glucose. Glycolysis technically is the breaking apart of glycogen, but the reason why I say glucose is because you start off with glycogen. If you remember, that's a storage form of carbohydrates in the body. Glycogen is basically, think of like a popcorn ball of glucose molecules, all stuck together. And so glycolysis is you're taking that glycogen, breaking it all the way down to the individual glucose molecule, and then that goes through the glycolytic pathway and then eventually the final step is pyruvate. Okay, that's referred to as glycolysis. The reason why people say fast and slow glycolysis is because glycolysis is an anaerobic system, so it works really, really fast. And it does give off a couple ATP depending on what you start with. If you start with glycogen, you give off 3 ATP. If you start with glucose, you give off 2. Remember, the end product, and we'll get through all of this right here, but I'm just kind of just bringing it up now. The end result of glycolysis working at a normal rate is going to be pyruvate, which then gets converted into acetyl-CoA, which goes into the Krebs cycle of the oxidative system. So people say slow glycolysis if glucose ends up as pyruvate, then acetyl-CoA, then into the oxidative system. Reason why that's not true is because the phosphagen system and glycolysis take place in the cytoplasm. The oxidative system takes place in the mitochondria. Okay, so once you get into the mitochondria, you cease being anaerobic, you start to become an aerobic system then at that point. Okay, so once glucose has become acetyl-CoA in the mitochondria, it starts then the oxidative phosphorylation process at that point. And the reason why I get so specific on this is because it'll start to help you to understand metabolism at a deeper level. And so for the sake of this class, I'll never refer to as fast or slow glycolysis. Okay, I mean, at times I may say that based upon the notes in the book, slip up and say something like that, but for the knowledge I want you to know is glycolysis takes place in the cytoplasm, and it's the breakdown of glucose or glycogen in the pyruvate. That's it. There is no fast or slow glycolysis. There's no anaerobic or aerobic glycolysis. That's by definition, glycolysis is, like I said, an anaerobic system. So it cannot be anaerobic and aerobic at the same time. It could turn into one another. That is, glycogen can be broken down in the anaerobic system and then transfer into the aerobic system. But there's a misnomer on, you know what I mean? So it can't be an anaerobic glycolysis, aerobic glycolysis, because glycolysis takes place in the cytoplasm without oxygen, but then it also can't be aerobic. So anyways, this is where it gets confusing, all that. And that's why I don't like using the term fast or slow anaerobic or aerobic glycolysis. Just know Phosphagen, the immune system, glycolysis takes place in the cytoplasm, oxidative takes place in the mitochondria. Big understanding of that. And I want you to know that because I want you to start understanding, like I said, these energy systems at a little bit deeper level. So you get into the phosphagen. It says provides ATP primarily for short-term, high-intensity activities. That is resistance training, sprinting, is active at the start of exercise regardless of intensity. So I should bring this up right now. All energy systems at all times are working. Okay, if you have an energy system not working at all, we refer to that as death. Okay, it's which energy system is taking over as a primary supplier of ATP. That's what we look at then in terms of like what's the primary system. Like we're talking about like type 2 fibers. They're high glycolytic, high power they have high amounts of phosphagen pathways and enzymes along with that. Okay, and it's suited, like I said, what energy system is suited for the activity, just like we talked about muscle fibers. We have the right fiber for the right activity. We have the right energy system for the right activity as well. So if you're talking about high energy or short-term, high-intensity, high-power movements, the phosphagen is supplying the ATP for that. Glycolytic and oxidative still working, just not to this certain extent. And the way this reaction works is you take ADP plus creatine phosphate, and then the enzyme creatine kinase, creatine phosphokinase, CPK, catalyzes the reaction into ATP and creatine. Takes the phosphate from the creatine phosphate, puts it onto the ADP to get you ATP. Okay, it says ATP stores. The body does not store enough ATP for exercise. And I brought that up kind of in metabolism a little bit, that we just don't have ATP. I brought that up in neuromuscular. ATP is just not floating around. We have to ATP is like money. Okay, you don't want to be driving down the road with $400 in your hands stuck out the window going as fast as you can because you probably lose a lot of that. ATP is kind of the same thing. And so how well we're able to take these nutrients, break them down to give you ATP, the better off you're going to be with that activity, with that energy system along with it. Can I bring this up? Those who have me for ex-phys heard this all the time. But when you're looking at these reactions, we deal with what's called rate. The rate of ATP breakdown versus the rate at which ATP is built up. Okay, the better, if you're able to build up faster than you're breaking down, then the body will stay within the energy system, keep energy going, that's keeping you going. But when you start to break down faster than you're building up, then you'll start to switch through different systems till you maintain that ratio. That's fatigue. That's what, when you start to fatigue, it takes place. Just think of when you go do, sprints, you can only sprint for a short amount of time before you start to slow down into a jog. Okay, that's where your energy systems are switching over because you can't maintain those energy demands to keep your body going during that activity. And so that's why when you're looking at metabolism, you have to look at how much substrate's available. Substrate for this system, the phosphagen system, is going to be creatine phosphate. What kind of enzymes? How well are the enzymes working? All that will help out with these reactions. So when you're dealing with the high intensity, short duration type activities, you're only going to last working at its fastest rate only about 20 seconds thereabouts. And I bring this up too, when you're trying to train, it's one thing to try and train athletes based around time, thinking you're going to hit the energy system, but really you need to look at how hard you're working within that time because rate will determine ATP turnover more than time will. Okay, a 30-second sprint is much different than a 30-second crawl. I'll burn up my ATP a lot faster in a 30-second sprint than crawling for 30 seconds. So you gotta be careful with time. You have to go off of rate before you go off of time. Now, when you're training athletes, if I'm having them run as hard as I can for 30 seconds, good chance, you are hitting either the phosphagen or the glycolytic system. But I'm bringing this up to kind of get you to start understanding how these systems actually work and operate. Okay, so ATP stores. You don't store enough, so you have to constantly keep replenishing it. And if you're able to replenish faster than you're breaking down, then you'll maintain in the whatever energy system you're in. So high intensity, short duration, you could only maintain that ratio for so long before it's going to break down because you're working at such a high rate, such a high intensity. You got to control the phosphagen system. Law of mass action. The concentrations of reactants or products or both in the solution will drive the direction of the reactions. Okay, and this just goes into what I was talking about with substrates. If you have enough substrates available, it'll keep the going. But what happens a lot of times is you run out of, especially the phosphagen system, you run out of substrate. That is, you run out of creatine phosphate. So therefore, you can't maintain ATP if you've run out of creatine phosphate. And that happens during the high intensity, short duration type activities. And that's why I say, I bring up the phosphagen system, but I really like to call it the immediate system because you're dealing with just an immediate reaction. So within this system, the short duration, high intensity energy system, we got the ATP, CP, the phosphagen system like we talked about. So you got ADP plus creatine phosphate, give you ATP and creatine. Well, creatine phosphokinase, by the way, is a rate limiting enzyme. So it's either going to speed up or slow down this reaction based upon demands. But as I said, you run out of substrate, the systems will slow down. You run out of creatine phosphate, well, this whole entire reaction will start to slow down. You'll start to switch to another one. Problem is, if you need ATP, you got to maintain energy somehow. So that's why we talk about the myokinase system. This enzyme, or the reaction here, is myokinase or adenylate kinase. And so what's happening with this part of the immediate system is you run out of creatine phosphate, you need ATP, but you have a high buildup of adenosine diphosphate. So what the myokinase system will do, will take two ADPs to give you an ATP and an AMP. Within the immediate system, the phosphagen system, there is no enzyme that takes an ADP plus an AMP to give you ATP. Because you could only have reactions occur if, one, you have the substrate available, or in two, you have the enzyme to catalyze the reaction. Within this system, there is no enzyme to catalyze the reaction of ATP and AMP to ATP. So the myokinase system will be able to go two ADPs to give you an ATP and AMP. The system also works backwards as well. So like during times of rest, the myokinase system will take the ATP and break it, or give an AMP, break them to get you then two ADPs again. Okay, but here's a problem. This is why fatigue sets in. Well, one, you run out of creatine phosphate. You can't match, number two, then you can't match ATP demands. But then three, you have a buildup of ammonia. So what happens is, as you start to break apart the ATPs, you get the ADPs built up, then the myokinase comes in, gives you the ATP and AMP. Well, then you have the buildup of the AMP. And the AMP does is then, via AMP deaminase, will then break it into inositol monophosphate and ammonia. I always say in my ex-phys course, if you use ammonia to strip the floor or use it to clean a toilet, it's not going to be good in the cell. Now, normally we could clear it out, but when it gets produced at high amounts, what we normally do is we flush it out to the kidneys and we excrete it out in our urine. But what happens is, well, during exercise, we have redirection of blood flow, so you have ammonia floating around, and it crosses a lot of different cell membranes, and the theory is they cause a lot of disruption in your neural activity. So ammonia is very caustic and is a fatiguing byproduct. And another problem here, too, is you went from an AMP, so adenosine monophosphate, to an IMP, inositol monophosphate. You've lost an adenosine group. So therefore, you've lost another ability to produce ATP. So there's a lot of problems when this starts to occur through here. That primarily happens in fast myosins. And in slow myosins, you have AMP get broken into adenosine and a phosphate. So once again, you're breaking apart and losing adenosine groups. So that's the immediate system or the phosphagen system. Then you have glycolysis. So the breakdown of either glycogen stored in the muscle or glucose delivered in the blood to resynthesize ATP. There's 12 steps to glycolysis. We don't need to know all of them. The biggest things you need to know is what's controlling glycolysis. And actually, the one thing that controls all of your energy systems for the most part is your adenosine diphosphate buildup, your ADP buildup. Because if you have the way metabolism works is this, there's a constant feedback system going on telling the body either speed up, slow down, or maintain what's going on. So if you have a buildup of ADP, but you need energy, that tells the body, start kicking up, start getting overdrive, we need some ATP. And if you have too much ATP built up, that's going to tell the body, well, if we don't need it, slow down, save the energy, we'll save it for later. For those who had me for X-Phys, I talk about the Augustus Gloop principle from Charlie in the Willy Wonka in the Chocolate Factory, where the kid falls into the chocolate river. His mom yells, Augustus, save some room for later. Okay, the body wants to make sure you save a little bit for later because you never know, you might need whatever it is later on. So when you're looking at glycolysis, just like immediate system, ADP is a big driver. Some other things that are important is NAD. So you got NAD reduced, and then you got the oxidized. So nicotine, amide, adenine dinucleotide. The proper ratio of that will also determine glycolysis working as well. Because you can see in the pathway you produce, what happens, you start off with glucose. You can see it's a 6-carbon molecule. And then you'll go down a few steps. You can see, well, one, you have to use ATP to get glucose into the cell. You see hexokinase. Hexokinase puts a phosphate on a glucose, but then it traps it in the cell as well. So 2 things: it activates it by putting the phosphate, and number 2, it traps it in the cell. And so from there, we'll continue on to then you go fructose 6-phosphate to fructose 1,6-bisphosphate. Phosphofructokinase, PFK, is a rate-limiting enzyme for glycolysis. It's either going to speed up or slow down the reactions. Okay, as it continues on the pathway, it'll split into 2-3 carbons, and then from there, you'll see NAD gets a hydrogen put onto NADH on both separate pathways right there. And if both go down, continue on to the end, and then you end up with pyruvate. Okay, why bring that up? Because in order for glycolysis to work properly, you need to have ADP, NAD+, and some hydrogens laying around. Because what happens is you take the hydrogens, put onto NADH, and then that allows for all these reactions to keep occurring throughout. So if there's not enough NADs within the cell, then glycolysis will stop and cease to keep running until you get some into the cell itself. And if there's too much NADHs, well, I'll get in that here in a sec, a little bit more, but if too much NADHs, that's going to get converted then into lactate. I'll talk about that here in a sec. Okay, NADH, where that goes to is, remember, back to the electron transport chain, which then helps out with ATP production. So what are you getting from glycolysis? Well, you're starting off with either glucose or glycogen. And going through the whole process, you'll end up then with, well, if you start with glucose, you'll get 2 ATP. If you start off with glycogen, you'll get 3 ATP. Glycogen, you get 1 extra ATPs because glycogen's already been phosphorylated. So therefore, you don't need to put a phosphate onto it to trap it in the cell because it's already done that. You also will get, assuming things keep working properly, you'll get 2 pyruvate and then 2 NADHs. All these are needed to make sure glycolysis keeps working at a proper rate. Because if not, what happens then is things start to get out of whack. And then you need to start clearing out. So like the example I use is, think of like a river flowing nice and easy. But what happens if, like, well, I'm from out west, and so I grew up in the mountains, and we have beaver dams everywhere. What do beavers do? most ranchers, most farmers, they'll know that beavers on property, sometimes the best, not the best, because they'll dam up a river and flood out different areas. Okay, and so think of a river kind of flowing through, and then what happens is a bunch of beavers show up and dam it up, and you have a buildup before the dam. That's what happens when glycolysis works at too fast a rate. Because the goal is, if you go back to here, once you've converted and produced pyruvate, pyruvate goes into the Krebs cycle. Krebs cycle starts working on oxidative phosphorylation. But if you have a buildup of too much pyruvate and too much NADHs because everything can't work at a proper rate, there's where you start to form lactate. Okay, so to go through it all right here, what you're going to end up with going through glycolysis, you're going to get 2 pyruvate, 2 to 3 ATP, and 2 NADHs. This right here says glucose, so you're starting off with, you got 2 ATP. That could be used, the ATP could be used wherever. So you got the end result of glycolysis is ideally pyruvate. Pyruvate will go in 2 different areas. There's 2 fates for pyruvate. If too much pyruvate is being produced, then you're going to have too much NADH as well. They're just going to go hand in hand. Buildup of pyruvate is going to generally go along with a buildup of NADH as well. What's going to happen is lactate dehydrogenase is going to take the hydrogens from the NADH and put it onto pyruvate. And then that forms then lactate. Another thing, a fate of pyruvate will be then converted into acetyl-CoA, which will then go into the mitochondria, which then go through the Krebs cycle. Okay, then it says ATP resynthesis rate is slower, but it can occur for a longer duration if the exercise intensity is low enough. So that's how we start to get a lot of ATP being produced, is by having those go in, having the pyruvate converted into acetyl-CoA in the mitochondria, and from there starts to then go through oxidative phosphorylation. Here's where they say the process is referred to as aerobic glycolysis or slow glycolysis, and that's a term I don't like using, because once you've gained into the Krebs cycle, you're now oxidative phosphorylation at that point. So I don't like using those terms. Okay, it says a formation of lactate from pyruvate is catalyzed by the enzyme lactate dehydrogenase. The end result is not lactic acid. Remember this though. Well, it's a isoform of lactic acid. So lactate is what we actually produce. The reason why lactate is being produced is because glycolysis is working at too fast of a rate. So you have a buildup of pyruvate and you have a buildup of NADHs. So lactate gets produced because it helps then to buffer out those hydrogens. Because remember, I said you take the hydrogen from the NADH, put it on the pyruvate, there's where we produce lactate. So what lactate's actually doing is it's actually buffering out the cell. It's pulling the hydrogens out, so therefore glycolysis can start working at a much more smooth rate, allowing for the end product pyruvate to go into the mitochondria that can start producing ATP via oxidative phosphorylation. So lactate does not cause fatigue. If anyone's ever told you in any of your classes that it does, now you can tell them that Dr. Farney says no, it doesn't. Okay, and then you can explain to them why lactate's beneficial. It's a buffer, helps out buffer out the cell. And in all actuality, what lactate really the body wants to use lactate for is it wants to use it for fuel, i.e. the heart uses it. The heart has special enzymes that could take lactate and convert it back into pyruvate, which they then go in the mitochondria for fuel. So lactate is good. It helps out. Now, it's associated with fatigue because in the old days, when they first started studying this, they saw as people tired out, lactate started being produced. So they thought lactate caused fatigue. No, there's association. That's just because as you're starting to fatigue out, well, you're having a metabolic disturbance in the cell. So then a byproduct, because when you ever have a disturbance, something else gets produced. And that's what's happening. The cell's not working quite as well because there's too much other stuff going on. So therefore, at that point, you have to clear it out and get it out. So that's why lactate is helping out, because it's helping to clear everything out at that point, especially during those high intensity type exercises. Okay, so just keep those in mind. Keep knowing that glycolysis to work properly, you need to have ADP, NAD, and of course the substrate glucose. If you've glycogen depleted, run out all glucose in the body, then glycolysis will just slow down big time. At that point, if you run out of glycogen and glucose, you're fatigued out at that point. Okay? And keep in mind, remember, I know the book talks about anaerobic glycolysis and aerobic glycolysis, but for me, those are incorrect because glycolysis takes place in the cytoplasm. When you get into the mitochondria, that becomes aerobic phosphorylation or oxidative phosphorylation at that point. So I won't use these terms outside this slide right here. And just remember why lactate forms. That glycolysis is working at too fast of a rate, so therefore you have to clear out the byproducts to get everything working back properly again. Lactate doesn't cause fatigue. Lactate helps then act as a buffer, clearing out the byproducts, but also lactate helps as an energy source for certain muscle fibers, like type 1 fibers, especially myocardium fibers. So I said that lactate can be converted back into fuel in certain fibers, like type 1 fibers, like myocardium, but lactate can also be converted back into glucose. The way the body does that is in the liver via gluconeogenesis. That is the production of glucose from a non-carbohydrate source. And the Cori cycle is that which takes lactate, transports it in the blood, then to the liver, and then from there, it'll get converted back into glucose. And so this is a way that we could keep producing glucose when we start to run out, primarily. We start forming new glucose this way to get transported out back to the muscle. Your red blood cells, for instance, they really rely upon this because red blood cells don't have mitochondria. So all the lactate being produced in red blood cells has to go somewhere. They'll go to the liver then to go through the Cori cycle to become glucose. So getting into then once glycolysis takes place, you produce pyruvate. Well, pyruvate will then go into the Krebs cycle. Once it enters into the mitochondria, it will be converted to acetyl-CoA. And at that point, then it'll start then initiation of the Krebs, which point of Krebs cycle is to get NADH, FADH, and ATP. And that goes, ATP is used for fuel, NADH and FADH will go to the electron transport chain to get produce more ATP. So things that do control or glycolysis, high amounts of ADP, phosphate, ammonia, and then it's inhibited by lower pH, ATP and creatine phosphate, citrate, and free fatty acids. Citrate is a main intermediate of the Krebs cycle. Also affected by hexokinase, which is the enzyme that converts glucose into glucose 6-phosphate. Talked about that, where it traps the glucose in the cell. Rate-limiting enzyme of phosphofructokinase and pyruvate kinase. Kind of finishing up then with lactate and you got the lactate threshold. So lactate threshold is basically at that point where the body becomes primarily anaerobic. So during exercise, as we're going along, we'll have the mixture of the energy systems going on. But once you reach a point where the body becomes solely anaerobic, that's where you start to see lactate start to skyrocket up. And that's the point called lactate threshold, where the body, you see the huge incline in lactate production. And it says represents an increase in reliance on anaerobic metabolism, also marker of anaerobic threshold. It says the intensity and relative intensity at which blood lactate begins to abrupt increase above baseline concentration. And then we talk about the onset of blood lactate accumulation, which is a secondary response. So initial response is lactate will start to rise when we start to exercise. And it says begins to increase significantly at 50 to 60% of maximal oxygen uptake. And that's in untrained individuals. For trained individuals, about 70 to 80%. So that's the initial response. Lactate starting to be produced then. And then you have the onset of blood lactate. It says a second increase in the rate of lactate accumulation. And it starts to get to a point to where eventually the body starts to become overwhelmed. This is where they thought lactate caused fatigue. It's just that lactate will rise along with intensity. And once you reach a certain point, your body becomes so anaerobic that just cannot maintain aerobic demands. And there's the association, not the causation. Here's just a look at the incremental increase. So with heart rate, you see heart rate increasing as intensity goes up. And the purple line then is the lactate response. So you start to see once you get to a certain intensity, lactate begins to start to really increase. But then once you hit the higher intensities, that's where the onset blood lactate really appears. And you see the vertical climb in lactate levels. And that's at the point where you're basically at pure, your highest level, your highest VO2, your maximum power output kind of deal. You're at that point where you're solely anaerobic. You just cannot match and maintain aerobic demands at that point. This can be pushed further to the right with training. So the more trained you become, the better off you're able to handle metabolically without having this huge appearance of lactate. So you can move this to the right with more aerobic training. And that's this is going to happen no matter who you are, the appearance of lactate. But to that certain point to where it becomes overwhelming, you could move that point, the threshold to the right with a lot more aerobic training that'll help out just to basically help metabolically handle all the processes before it becomes overwhelmed. Getting into the oxidative system, this is the primary source of ATP at rest and during low intensity activities. So this is really what keeps us alive. Aerobic metabolism, oxidative phosphorylation, that's really what keeps humans alive. I mean, that's the basis of life right there, cellular respiration. This is the primary energy system at rest and during low intensity. It doesn't take much, though, for you start to become more glycolytic. Okay? It doesn't take much more away from rest where you start to have some kind of switchings taking place. The benefit of the aerobic system is it can use carbohydrates, fats. It can also be used protein for fuel as well. So it can use all sources within it as a substrate. Obviously, protein is not ideal, but it can help out if given that you need to. So first you get glucose and glycogen oxidation. This is when you have glucose glycogen broken down into pyruvate via the Krebs or glycolysis. And then from there, enters in the acetylcholine into the Krebs cycle. And I just put their terms right there, the TCA. So the tricarboxylic acid cycle or the citric acid cycle. Those are their terms. I like using Krebs. It says the FADH and NADH transport hydrogen items to the electron transport train where ATP is produced from ADP. We'll get into that coming up. So the major parts of Krebs is, one, you have the coenzyme A, and what happens is pyruvate gets converted into or reacts with the coenzyme A, become acetylcholine. In the start off Krebs cycle, those who have me for XFIZ probably got tired of me talking about this, but the things needed to start Krebs cycle is you see acetylcholine, but that needs to react with oxaloacetate. Those working together form citrate, hence why it's called the citric acid cycle. But acetylcholine plus oxaloacetate starts the cycle and keeps it going. Reason why I harp on oxaloacetate is because that comes from two sources, proteins and carbohydrates. You get it via carbohydrates via pyruvate being converted into oxaloacetate, or you break down other proteins in the amino acids, and then those get produced and turned into oxaloacetate. So the goal with this cycle is you want to use as much carbohydrates as possible. Okay, and so everyone always talks about like, well, I'll just use my fat as fuel. That's true to a certain point. But you always need, it's just, when you're using fat for fuel, it's just not fat you're using. If you want to use fat, because what happens if you go back to beta oxidation, what's happening with our lipid oxidation is that you're taking, we'll get into this coming up here, but you're taking a triglyceride and breaking it down into acetylcholase. Okay, so you get acetylcholase coming into the Krebs here, which is fine, but you need oxaloacetate to get the whole cycle going. So if you just have the fat or the acetylcholase from fats, you need a second part. That's why we always say fat always burns in the carbohydrate flame, because, and ideally to burn as much fat as possible, you want to have oxaloacetate coming from then carbohydrates to keep everything going. If you're turning proteins to oxaloacetate, you're glycogen depleted at that point. In terms of performance, you're finishing not first. Okay, if you become severely glycogen depleted, you're not going to be on the podium probably at all. So glycogen is so important for fat metabolism, protein metabolism, and of course, carbohydrate metabolism as well. Okay, proteins you can see on here, they're used to make all the different intermediates. So anything you see the words, those are all the intermediates. Like citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. That's how protein metabolism is you're not really actually taking protein and making it into, or breaking it down for ATP. You're breaking it down into amino acids, which can be used for intermediates. Okay, and so what Krebs, remember, every time Krebs turn, it comes from, if you have one pyruvate, you'll have one turn of the Krebs. Normally from glucose, we'll have one glucose turn into two pyruvates. So therefore, we get two turns from the Krebs. So we get a little bit more. If you're having sources coming from carbohydrates, from glucose or glycogen, you get a little bit more ATP this way. Because for every turn of Krebs, you're going to get one FADH, which you see right here, flavin, adenine dinucleotide. You get 3 NADHs. Remember, NADH is nicotine amide adenine dinucleotide. And you get one ATP. Well, it says guanine triphosphate, and ATP and GTP are the same thing. And so the GTP can then be used for energy wherever ATP is needed. The FADH and NADH will then go to the electron transport chain. We'll see that here in a sec. Okay, so just remember the basis of metabolism here. I know those who had me have heard all this, but those who didn't have me for XFizz, remember what I keep talking about, why I harp on oxaloacetate. Okay, if you don't remember that word long-term, what I want you to take away from is, in order to keep performing at a certain level, you need to have enough glycogen. That's the take-home message. Okay, even if you want to burn up fats, you need glycogen to help out with that. You never want to self-cannibalize and start eating protein away. That's what happens when you become glycogen depleted. So even those people who are on, big time, intermittent fasting, I think is a good way to go about, but you got to be careful. Sometimes those who lose a tremendous amount of weight and they feel good about themselves, okay, they have lost weight, so that's good. But you got to be careful in terms of performance, because if you've lost a significant amount of weight while reducing carbohydrates big time, which is what a lot of those people are doing, well, you're actually probably in turn burning a lot more muscle than you are fat. The scale is reflecting losing weight, which is, don't get me wrong, that's a good thing. But if you're burning up a lot of protein, that's not necessarily a good thing kind of in the long run. And really, it's about, if you're glycogen depleted, it's about a 50-50 ratio between fatty acid usage and amino acids. So that's never a favorable situation to be in, whether you're performing for competition or you're just trying to stay healthy and lose weight. Okay, sometimes people panic about carbohydrate intake. Well, yes, too much carbs will add to weight gain, but so will anything else. And sometimes it's not what you eat, but it's when you eat too. Okay, not eating carbohydrates could be just as bad as sometimes eating too much. That's why it's more beneficial, in my opinion, to, if you're going to work out at late at night, eat most of your carbs around that time. Don't eat them all day long. Or vice versa in the morning, don't eat them the rest of the day after you train. We'll get into that in the next units with nutrient intake. But the take-home message here is, if you want to keep operating, performing at a level, which is what this class is about, performance, make sure you have enough glycogen coming in. That'll supply the oxaloacetate demands. We never burn up all our fat, because fat's always, all of us are going to have enough fat. So that can supply those demands. You never want to turn in the amino acids for fuel. So remember these things I talked about, these important parts, because I'll test you on them, but also because this is starting to get in your mind, like understanding how all this is starting to work, because this will carry this into nutrition and eating them for performance. And here's electron transport chain. The sole purpose of this is to produce ATP in water. Okay, the NADH, FADH, they show up, they drop off the hydrogens. Hydrogen is one electron, one proton. So the electrons leave the hydrogen, hence why it's called electron transport chain. The electrons go through a series of reactions, and then that helps produce then ATP. And then the hydrogens meet up with oxygen, give us water. So the ETC's purpose is to make ATP and give us water. And you got coenzyme Qs, which are just proteins within here that are important for all these reactions. And then cytochromes are these are all just important parts that keep the ETC going. And so sum it up, NADH, FADH bring in the hydrogens. Therefore, the purpose of ETC then is to take those hydrogens, produce water and ATP. Right-laning the enzyme of the aerobic system is going to be isocitrate dehydrogenase. It says it's stimulated by ADP and inhibited by ATP. That means as ADP builds up, it starts to speed up. As ATP builds up, it'll start to slow down. It says the rate of the Krebs cycle is reduced if NAD and FADH are not available in sufficient quantities to accept hydrogens. So those are important coenzymes that need to be around to keep everything functioning. And the ETC is stimulated by ADP and inhibited by ATP, just like with the Krebs. So fat oxidation, start off with triglycerides, which are 3 fatty acids on a glycerol backbone. So it's stored in fat cells and broken down via hormone-sensitive lipase. This releases free fatty acids from the fat cells in the blood, where they can circulate and enter muscle fibers. Some do come from intermuscular sources, and that's usually what we talk about with athletes called the athlete's paradox, where athletes are able to store fat within muscle cells, which means you don't have to transport fats to get them to where ATP is needed. Problem is most athletes are super lean, so they can store fat within the muscle, which is on site. Problem is most athletes are too lean, so they can't get to the point where they store because they use it up. It says free fatty acids enter the mitochondria, are broken down and form acetyl-CoA and hydrogen protons. The acetyl-CoA enters the Krebs. The hydrogens are carried by NADH and FADH to the electron transport chain. Protein is not a significant source of energy for most activities, and that's true. We don't want to use protein. But there's a direct relationship between glycogen levels and protein oxidation. That is, as glycogen gets depleted, protein oxidation increases. So if you want to use protein for fuel, get glycogen depleted. If you want to burn up your muscle, have zero glycogen in the body. We break down protein into amino acids, which then can be used as a different intermediates within the Krebs cycle. Also converted in pyruvate and also can be converted into glucose. But it takes a long time and it's very costly to do that. So ideally, you never want to be in that situation. Okay, and here's just kind of the overall pathways. There's some common ones, so such as the good thing about the oxidative system is they get used fuel sources from, like I said, fatty acids, carbohydrates, and proteins. And so they're all reduced down to acetyl-CoA, enter into the Krebs. You all use ketones as well. Once again, if you're using ketone bodies, that's due in part because you're glycogen depleted and you're having to produce ketones, which then could go into the Krebs itself. Too much amino acid breakdown, given off as urea. So that's one way to tell protein breakdown is via the urine. But the goal is here to have all this operating functioning like a river, as fluid as possible. Okay, buildup anywhere is going to have a backflow and affect all energy systems. And usually we see that with untrained. Untrained people just doesn't take long before metabolism becomes disrupted. So you want to make sure everything's flowing as properly and easily as possible. And just remember the relationships I talked about in terms of glycogen levels, the byproducts or what you get produced from glycolysis. Okay, pyruvate, lactate, oxaloacetate, acetyl-CoA, amino acids, all these things coming in, because I'll talk about these as the lectures go on, and I'm going to assume and know that y'all know this, okay? And so whatever you had in other classes, if there's a kind of disruption or confusion, please email me and I'll explain to the best of my abilities. Especially I know terms like fast glycolysis and slow glycolysis. Okay, I might confuse y'all, but I hope what I went through, what I explained, that makes sense to y'all now. So just know the main parts. If you can look at this slide and understand basically the overall parts of all this, then you'll be good. Okay, one last thing I just realized on here too, bring up with triglycerides. Ideally, during most exercises, glycogen levels going to, let me take that back. Glycogen is going to determine pretty much overall metabolism during performance. Triglycerides, ideally we want to use them, but there's a trade-off. It takes a while to use triglycerides for fuel. The oxidase system is a reactive system. It takes forever to get going. Okay, it takes a while to break down the triglycerides into the fatty acids and the glycerol. Glycerol can enter into glycolysis. You can see it right there going into phosphoglyceraldehyde. And then from there, it goes through the glycolytic pathway. So you get a few more ATP by using glycerol along with the free fatty acids. But in terms of performance, glycogen is going to dictate it all. It really is an important driver for overall metabolism during performance. Okay, so just to kind of rehash on all this, I hope if you can understand all these parts we talked about, then you'll know what you need to know for moving on as a strength coach or working with athletes or training anyone in general. Okay, you'll understand the relevant importance of glycogen levels and overall performance, about protein breakdown, so forth. Okay, I'm not going to get too concerned on how much ATP exactly for, it says like one glucose molecule, because there's a variance. Okay, we're not really too sure exactly what it is. I mean, there's a lot of different numbers that come about with all this. Okay, but it says in general, there is an inverse relationship between a given energy system's maximum rate of ATP production and the total amount of ATP is capable of producing over time. Okay, what we're getting at with this is, I brought this up earlier in the lectures, but at the bottom side talks about time for energy systems. What I want you to know is with energy and substrate utilization, it's all based upon rate first. How fast the energy system or how the rate at which ATP is being produced versus breaking down will then determine how long I could actually be sustained in that energy system. Okay, like we said, we know the phosphagen system is a super high intensity, high power system. So if it's working at its fastest rate, it can only go a short amount of time. On here it says 0 to 6 seconds, really up to 30 seconds, you could use the phosphagen to some extent. Okay, that means that at its fastest rate, the phosphagen system will be the primary energy system for only about, less than 30 seconds, probably less than 20 seconds. Key, though, is at its fastest rate, working at its maximum rate. Just like glycolysis. I know it says fast and slow in here, but please, you know what are my thoughts on that. Glycolysis really only could work for at its fastest rate overall, really only about a minute. Okay, it says 30 seconds, 2 minutes here, but you see a lot of older studies, once you get past that minute, you start to get a little bit more oxidative if you're still able to keep working at that same level. And so really, there's a lot more oxidative taking place than probably think of at that time frame. So the point I'm getting at this is what's driving what energy system is, first of all, how fast that system is working. And then from that point, to kind of determine how long you can stay within that. So now if you're training athletes, it's your goal as a coach to try and get athletes to perform as hard as best they possibly can within a time frame. That is, if I'm doing repeat 110-yard sprints, I know I should take them, depending on the athlete, less than 15 seconds. Even less. Some of the alignment, sometimes I think I've seen it maybe a little bit, 17, 18 seconds, something like that. I'm not sure what all coaches have on those time, but let's say less than 20 seconds. I know as a coach, if I'm going to make you sprint, do repeat 110s, I'm trying to push you to run as hard as you possibly can within that minute. That's my goal, or within those 20 seconds. That's my goal. So ideally, you're trying to, within that time frame, you give them a time frame, but you're pushing the work as hard as they possibly can. So there's where you're starting to get the adaptations, the training to the energy system itself. Okay, and like it says, the extent to which each of these energy systems contribute to ATP production depends on the intensity of muscle activity and secondary on the duration. Okay, so that's why I keep saying, just remember that. It's all based upon the intensity, the rate of what you're doing, how hard you're working. Okay, no energy system provides all their energy at once. It's a contribution. The harder you work, the more phosphagen and glycolysis contribute. The longer you work, the more oxidase system is going to contribute. Okay, so just remember on all that right there. You got depletion and then repletion. So phosphagen, so creatine phosphate, it decreases markedly, within the start of exercise. It says within the first stage, so 5 to 30 seconds of high intensity, almost all of your creatine phosphate will be depleted. However, a good thing is it can be repleted relatively quick. It says complete resynthesis of ATP appears to occur within 3 to 5 minutes and complete creatine within 8 minutes. Now, most places will even say within about 5 minutes you start to see creatine phosphate start to be fully repleted as well. So it's a good thing to know if you're doing repeat work, you're working on power movements, doing repeat high intensity type work bouts, to make sure you give yourself ample amount of times. That way when you perform the second bout, you can perform almost at the same level, due in part by having the creatine phosphate being replenished. Glycogen says depletion is related to exercise intensity. It says that relative intensities of exercise, so above 60% of maximal oxygen uptake, muscle glycogen becomes an increasingly important energy substrate. The entire glycogen content of some muscle cells can become depleted during exercise. And rarely, though, if you look at a lot of studies, within about 30 minutes, almost all at a relative intensity, like it says, about 60%. So that's usually a moderate relative intensity right there. Pretty much all your glycogen can start to become depleted that fast. And 60%, for those who are trained, 60% is really not, that's like a decent jog. Okay, for those who are trained, that's really not that much. And so at that point, the harder you work, the sharper the decline in glycogen. And there's a direct relationship between glycogen levels and performance. As glycogen goes down, performance will decrease. So it's so important that, like I said, in terms of performance, it's what you eat, but also when you eat becomes important too. Making sure you have enough carbohydrate storage, glycogen storage going into competition. That's why it's important during exercise too to be consuming some, and then post-exercise as well. Okay, it says repletion during recovery is related to post-exercise carbohydrate ingestion. So it appears to be optimal. 0.7 to 3 grams of carbohydrates per kilogram is ingested every 2 hours following exercise. Okay, 3 grams of carbohydrate per kilogram. Someone who weighs 220 pounds, that's about 100 kilos. So that's 300 grams ingested every 2 hours, assuming you came from a super high intensity workout. Okay, so just remember how important glycogen is for overall performance. The recovery period is going to also determine a bunch of other variables too within the human body. So we get into EPOC, so excess post-exercise oxygen consumption. This is essentially the time after exercise that it takes to get the body back to normal. And this is where the oxidase system is really ramped up, helping to bring all these all these processes, so like heart rate, temperature, creatine phosphate levels, glycogen levels, hormone levels, everything back to normal. So the oxidase system post-exercise is ramped up, and it's helping to bring everything back down to your pre-exercise condition. Okay, the old term was oxygen debt, but now we use EPOC. And that EPOC time, how long it takes to get back, is going to be determined upon the intensity at which you worked. And so this is low intensity, steady state exercise. You can see steady state, we have the oxygen deficit, which is at the start of exercise. Oxygen deficit is kind of misleading because we're never really deficient of oxygen. It's just the time where the requirements for exercise far outweigh the available amount. So when I start to exercise, it takes time. Let me take that back. The oxidase system is a reactive system. It's a slow system. It takes a while for it to get warmed up and get going. So when I start to exercise, my body, the available amount of O2 is far lower than the requirements for exercise. So that's the time where the glycolytic and your immediate system, your phosphocreatine system is really super ramped up, trying to get the body to get to match those O2 demands. Once the oxidase system gets ramped up and once it starts to take over, then at that point you have a greater amount of O2 available than the required amount for exercise for oxygen. That's referred to as steady states, when everything balances out. And so for low intensity steady state exercise, the epoch time is not going to be nearly as long as if you did a high intensity non-steady state type exercise. Okay, glycogen replenishment could take up to 48 hours after a super high intensity long, bout of exercise. And what high intensity, this is looking at right here, why you don't get into steady state is because the requirements for exercise are far greater than the VO2 demands. So this is your super high intensity type work where the O2 requirements to perform that work is far greater than your VO2, your actual available amount. So therefore, you never actually get in a steady state. And then from here, your epoch is going to be extended out big time because it's just a lot more stress on the body. So it's going to take a lot longer for the oxidative system to bring everything back down to normal, to bring it back down to pre-exercise conditions. Here's just a ranking of importance for different exercises and just a relative contribution. So pretty much falls in line, makes sense from everything we've been talking about here. It says liver glycogen on here. Reason why that's put up because the liver is basically a reserve tank for glycogen storage. So you run out of muscle, you could turn the liver as a reserve tank. Basically, you could think of it like that. You got specificity, so you got interval, and then high intensity and combination training. So of course, interval training is kind of it says emphasizes bioenergetic adaptations for more efficient energy transfer within the metabolic pathways by using predetermined intervals of exercise and rest periods. So interval training is just really, you're just combining different overall build up the energy systems to where you can transfer between them a lot more efficiently. Then you got HIIT training, which everyone's all about, which I do like HIIT. There's a lot of benefits from high intensity interval training. And so this is really, you're having this high amount of work with very short rest periods. They recommend the 1-to-1 work to rest ratio. And this is definitely over 90% of VO2 max. So this is super high intensity. Problem with this though, if you're doing a lot of HIIT training, but you're also training other ways, it does put a lot of stress. So you could increase the chance of injury. So you got to be careful of programming out your workouts on here. And in combinations, it says adds aerobic endurance training to training of anaerobic athletes in order to enhance recovery. May reduce anaerobic performance capabilities, partly high strength, high power performance, and can reduce the gain of muscle, strength, and speed and power related performance. So when you're looking at specificity, you need to look at essentially what it is the demands of your sports are. So if you have a power athlete like a weightlifter, they should do zero aerobic work because there's no point in that. And vice versa if you're an endurance athlete. If you're a power athlete, for instance, or you really rely upon your type 2 fibers, which we talked about in the last chapter, but type 2 have high amounts of calcium released or high SR complexes, a lot of calcium, therefore a lot of calcium pumps, which help pump out the calcium. But also when you do a lot of endurance work, you reduce the number of calcium pumps. So those will then in turn affect how well your type 2 fibers are working. Okay, so you need some sports combination is good. Soccer is a good sport where there's a lot of A lot of aerobic work mixed with a lot of anaerobic work. So there's something you could do, some need to be doing. Because really, the stats are on soccer match, they run between, I think, around 7 miles on average per soccer match. Okay, so that's a lot of aerobic work mixed with then high intensity sprints. Football, on the other hand, the average play is like 6 seconds, 5 yards, something like that. Okay, there's no point in having athletes like the old days used to make us go out and run miles to get in shape for football. There's no point in that. That's not going to help out. Best thing in shape for football is doing a lot of high intensity sprints. Okay, so that's where your specificity training is starting to understand the demands of your athletes, starting to understand what exactly do they need. The metabolic demands, the neuromuscular demands, cardiovascular demands. Okay, and that's what makes a good strength coach. Okay, when I was at LSU, I worked the 2014-15 season, 2014-15 season. And I got to work with Alex Bregman that year. He was at LSU his senior year. And the strength coach was a guy named Jeremy Phillips, who we called Truck. He was 6'5", 300-some pounds. He played offensive line at Georgia Tech, hence the nickname Truck. Okay, he never played baseball at all. Except the only times around baseball is when he was maybe like a little kid, but for the most part, just when he was a strength coach at LSU. Okay, but what he became, I started talking to him, he really, to me, is one of the best strength coaches I've been around because he took time to read about his athletes. A lot of times you get these strength coaches who've done the sport and they think they know all about the sport just because they grew up doing it, which, yes, there's a lot of truth that the more you do it, the more you understand the demands. But to be a good strength coach takes time to really learn your athletes. And Truck was a good strength coach because he read about baseball. He tried to learn about what actually, because there's a difference in training baseball players than football, than soccer, than gymnasts, and so forth. So when you're looking at training athletes, especially metabolically, learn the demands. What are the actual importance of the sport? Okay, baseball, for instance. You're running as a base runner, all you're running the longest, well, I guess inside the park run won't be the longest as a base runner you can actually do. But for the most part, you're looking at trying to be fast from first to second from home to first or home to a second. That's kind of usually the majority of your hits going to be within that. And I don't know the stats of actually percentage of all the hits, but for the most part, you've got to run to first once you hit it. Okay, if you get lucky enough to hit to, get a triple, that's a little bit further. But you know what I mean? Like as a base runner, you're running a very short distance. So in terms of what we talked about metabolically, what do you need to build up? A lot of creatine phosphate. Okay, even as an outfielder. Okay, center fielder is the one who has the sole power in the outfield compared to right and left field. So if the center fielder has got a better read on the ball, he takes over and will get over the left or right fielder. So center fielder has a lot more to run than anyone else. Okay, so he might need a little bit longer sprints compared to, or your outfielders in general will run more than your infielders. So they might need a little bit different training than your infielders. But for the most part, baseball is a sport where you're really not running that far. So a lot of creatine phosphate will benefit that sport. High intensity sprints. Okay, a lot of work like that. That's going to help out them. As opposed to metabolically like a soccer player who on average runs 7 miles per match. Okay, vastly different athletes, vastly different demands. So it's up to the strength coach to look at the demands of the athlete. What are the metabolic costs and how do I improve that? And just because you've done the sport, don't think that you actually know all about that sport. Understanding X-Phys is one thing. Having to teach you all X-Phys and neuromuscular and everything else is a totally different thing. I have to understand the material at a different level by teaching it than I did when I was a student taking the courses. Okay? So just kind of keep that in mind, as you learn about metabolism, how to train athletes and so forth. Okay, and this is just the key point right here. Just look at this, just kind of looking at the just everything we've talked about throughout all this, looking at the energy systems, the amount of power that is, And then like the rate they could be. So like your phosphagen system is a high power system. Glycolysis is, medium power. Oxidative is very low power, but it's also endurance. Okay, and just kind of understand some of these. And it also says right here the work to rest ratios. If you're doing high intensity sprints, you want to give yourself a little bit more time to rest. That way, help get everything replenished. So because if you're doing these repeat work, you want to be able to keep working at the same level. So that's why rests are beneficial at times. A lot of times, actually, It says the use of appropriate exercise intensities and rest intervals allows for a selection of specific energy systems during training and results in more efficient and productive regimens for specific athletic events with various metabolic demands. To conclude with this chapter, make sure you know the definitions and the major parts of all the different energy systems. Okay, I know I covered a lot of stuff that those who have not had me with probably a different spin, a different take on metabolism, but for the sake of what I want you to know is what I just discussed. Make sure you know that, we have the three energy systems, but what's controlling them is the rate at which these reactions are occurring. The main reaction is ATP hydrolysis versus ATP synthesis. How fast are you breaking down versus building up? That's what's really going to drive overall metabolism. If you're able to maintain that rate, then you'll stay within that energy system. You'll be able to ideally keep performing at that same rate. Once you switch the ratio and start breaking down faster than you're building up, that's when fatigue starts setting in. You start to switch between all the different energy systems at that point. Okay, so the phosphagen system, the immediate system, can only work so long compared to the glycolytic. Glycolytic only so long until oxidative. And really, in a lot of ways, once you move past rest, you start to slowly get into glycolysis faster, start using glycogen up faster than fat. And it doesn't take much for it to start occurring. Ideally, you want to be able to maintain as much lipids for fuel as possible, but it's what we call a crossover concept, where at a certain point, you switch from fat as a primary fuel source, substrate, to then carbohydrate. Those who are trained are able to use fats a lot longer into the workout than those who are untrained. But even those who are trained will eventually switch over and start becoming more carbohydrate dependent. And really, when you look at the crossover, it doesn't really take much past really being at rest and really low intensity workout exercise. Okay? Remember also the importance of glycogen. Okay, as glycogen goes down, performance will go down. As glycogen gets depleted, protein oxidation gets increased. Remember why glycogen is important. Oxaloacetate within Krebs cycle. Understand the parts of glycolysis, the end products, pyruvate, NADH, ATP. Why is lactate formed? The parts of the immediate, creatine phosphate, the myokinase, the oxidative system. Okay, I want you all to understand this at a decent level, because then it's really going to impact then how you train athletes. To be a good strength coach, you don't have to know every little intermediate. You don't have to understand every single part of metabolism. But to be a good strength coach, you need to understand the overall concepts. I will say the better you know this, the better coach you will be. The better you understand the relationship of the energy systems with ATP production, first the breakdown of it, the switching of the systems, the intensity of which you go work, all that, the more efficient of a coach you will be. Too many times coaches train athletes without understanding the needs of those athletes. Okay, growing up, probably most people my age and older, for football is always go out and run bunch of miles, get in shape for football. Okay, never do you run a mile in a football game. Okay, like I said, on average, most plays are a couple seconds, a couple yards. So why run a mile? Doing sprints would be better off suited for getting into the conditioning for football than going out and running miles. A professor of mine used to intern at the Olympic Training Center. in Colorado Springs, and that's where the USA Wrestling was. And so the wrestling coaches always have their wrestlers go in the weight room and do a bunch of metabolic, I guess, conditioning, high intensity work, interval training in there. Well, during practice, you're getting plenty of that. So in the weight room, why go do more of that? So in the weight room, when you think about the demands of a wrestler, you need to be strong. You need to be able to be, yes, conditioning. You need to be able to have the metabolic enzymes, the processes, pathways to supply the ATP for your event, but you also have to be strong. So if you're doing practice where you're building up the endurance part there, then the weight room you should be throwing. If you compete in the 85 kilo class, you should be in the weight room throwing an 85 kilo bar around. Okay, that's the specificity we're talking about. And to understand specificity, you have to understand the demands of the competition. You need to understand the demands of whatever it is that your athletes are going through. And to be a good strength coach, you need to know that how you train a football player is different than baseball, than basketball, than soccer, than gymnastics, volleyball, to golf, and so forth. Okay, so that's why I harp on so much all this, and I really discuss as much as I can, because there's a lot that goes into understanding how to make athletes perform. And the better you understand these processes, these pathways, and how they all work together, the better off you're going to be. You know, even if you don't want to be a strength coach and you want to, be a full-time sport coach at a high school, guarantee you're probably going to be some strength and conditioning at some point. And just think, even if you don't want to go purely in strength and conditioning, any coach who knows these processes better, no matter what position of a coach you are, the better you will suit your athletes. Okay? So, go through this all. Let me know if there's any questions. Email me, but go through these processes and go with what I've been talking about and start to try and paint these, look at what I'm saying about how they're related to one another, all the parts, and how the relationships all work together.